Good evening, everyone, and welcome to our latest STEMtastic pop-up session. You're joining us tonight for a masterclass in microscopy with Marty Jopson. If you have any questions for Marty, then pop them in the chat box, which you'll find in the bottom left-hand corner of your screen, and he'll do his best to answer them at the end of the session. Now that just leaves me then to introduce you to Marty and leave you in his very capable hands. Hi, Marty.
My name is Marty Jopson. Just zoom in. It's amazing, isn't it? Beautiful detail. Hello, everyone. My name is Marty Jopson, and welcome to my kitchen. Quite frankly, I'd love to have been down there with you in Suffolk, but 'tis not to be. So we'll do the next best thing, and I shall present to you through the Ethernet. I'm a science communicator. I've spent my career talking to people about science, but I started my scientific journey as a research scientist at the John Innes Centre in just outside Norwich, in fact. And there I trained, amongst other things, how to use these wonderful pieces of kit here. Well, not these particular ones, but you know what I mean. So these obviously are microscopes. Now I am actually, I am sponsored by the lovely people at Zeiss, these guys over here, and they give me these microscopes and tell me to go out, tell people the wonders of microscopy, spread the news, evangelize. So that's what I do. So let me take you on a bit of a microscopic safari, and let's do a little bit of microscopy, and I'll show you what we can see. So join me down at the desk. I'll press that button there, and here I am, second camera, a little bit closer up. So, okay, let's look at some things under the microscope. The first thing we're going to look at, I'm going to show you a variety of specimens, and I'm going to rattle through them at quite a pace. And I'm going to try and put in as much sort of science that's appropriate for you guys doing A-level biology. So any questions? In the chat window, if I notice them, I've got the chat open on my screen here. If I notice them, I will answer them straight away. If not, hopefully I can get to them at the end. And you can ask me anything you want about the stuff I'm looking at, and I will try and answer. So, okay, the specimen I'm going to show you first of all is under this microscope here. Now, this microscope here is, oh, there it is. This is a special type of microscope. There it is. A special type of microscope called a stereo microscope, not just because it's got two lenses there, but because actually what I see is it sort of looks in stereo. It looks like three-dimensional. It's really cool when you look down the lenses here. You don't get that when you look at the camera. That's the camera there. And the sample goes down there, and you can see this allows me to put a massive sample in because of the nature of the lenses. It used to be called a dissecting microscope, but they don't call it that anymore. Oh no. Right, back to me. So, okay, let's show you what's under the microscope at the moment. Here it is. This thing under the microscope, it's an insect. It's an insect. It's called Pediculus humanus capitis. And if you know any Latin, you should be able to work out what it is. And just by looking at it, you might be able to work out what it is. It's about 1.5 millimeters long. It's pretty common. Pediculus, skin, humanus, human, capitis, head. It's a head louse, obviously. These things are parasites on humans. They are obligate parasites. They can only live on humans. And that's one of the beauties of microscopy, because it allows us to see the detail that explains how these organisms live and go about their daily lives, so to speak. It is the tool of the biologist in the same way that, I don't know, the test tube is the tool of the chemist. That's putting chemists down a bit. But the biologist uses the microscope, and it is the biologist's tool. The thing is, if you look at Pediculus humanus capitis, the head louse, you'll notice, and I'm showing you actual microscopic, I mean, not showing you films I did sort of 2 days ago. I'm going to show you actual specimens right here, right now. Everything I'm going to do is live microscopy, so that you get a feeling for what it's actually like using a sort of a decent microscope. Because I'm suspecting that whilst you might have microscopes at school, probably not quite as fancy as these ones. So this, I've zoomed in as far as I can. I'm at ×5 here and ×10 here. So it's about ×50 magnification. But obviously, what you really need to know is how big is this thing that I'm showing you. And I'm telling you, it's about 0.5 millimeter across vertically, and longitudinally it's about 1.5 millimeters, 2 millimeters. But what you really need to look at are the claws. It has one on every leg, okay? And these are fixed claws. They're hooks like this. They do not move. Turn the sound down. They do not move, but they have a little kind of flippy bit here. Can you see that? There's a little sort of flippy bit that sticks there. And the key thing about these hooks is the diameter of that hook there is the same as the diameter of a human head hair. And these little critters will only live on your head. They don't live on hair anywhere else on the body, right? Nowhere else, because all the other hair on your body is of a different diameter, and they can't hang on to the hair on those, so they slip off too easy. Okay, so they can't go anywhere else. So there are different head lice for different parts of the human body. These guys are completely harmless. The ones that you get on your body louse, which is a different type of, I think it's Pediculus humanus corporis, lives on the body, corporis, the body, and that hangs onto the tiny hairs, these tiny hairs that you get on the backs of your hands and things like that. It's got much finer claws. So that using the microscope and the power of something like this, you can see more information about a creature, a little creature like this. And that gives you some information about what it has evolved to do. And this thing has evolved to live on the human head, nowhere else. Other animals' hair tends to be much thicker, so it doesn't survive on cats or dogs or other domestic animals. One of the nice things I like to show you, and this is actually one that I combed out of my own hair after one of my children came back with head lice and gave it to me, and there it is. You can also, if I zoom out, where is it? I'm just going to, and I just to prove that I am actually showing you here. Here's a, oh look, they're slightly on top of each other. There are two things there. Let me see if I can just push them apart. They've got tangled up. Let's push them apart a little bit. That's better. So you've got two things there. At the bottom, immediately below me, is the nit, the egg, and this is the same magnification, mind you, that I've got it on now. And that's that female, because 'twas a female, laid that egg, which makes your eyes kind of, that must have smart. And this thing here is actually a bit of my hair. And can you see at the bottom of the egg, just there, there's a little sort of triangle of goo, and that is all that remains of the cement that the female secretes once she's laid the egg. And the cement then wraps itself around the hair and onto the egg and sticks the egg to the hair shaft. And that's a bit of my hair there that I combed out. And that is what would have hatched out. That is a first instar. It goes through, I think it's 3 instars, 3 molts before it becomes a full-grown adult. And then it will just plow around on your head and nibble, bite your head, suck your blood. It eats blood, of course, and lays eggs. So there's a delightful little thing to show people. One of my own head lice. What fun. Okay, let's look at something else. Let's look at something a little bit bigger. This is a critter that I pulled out of the back of my garden just now. And I just wanted to, I'm just going to show you this very quickly because this is something I only discovered. And the more you look down the microscope at things, the more amazing things you discover. It is, of course, a spider. This is a spider. Some people really hate spiders, and I have had people when I've done live performances of this that have had to leave the room and screams and stuff like this, which, as far as I'm concerned, is a result. This is a spider. It's called Tenegaria Domestica. This is the common house spider. It's not the really monster one. That's Tenegaria Gigantia. It's just the big one. You're getting a lot of them coming into the house at the moment. This is a female. You can tell it's a female by the size of its abdomen, by the shape of its abdomen. The males have a kind of a straight abdomen. It doesn't kind of curve out like that because obviously they don't need to lay eggs. But you can see a lot of detail. I'm going to show you one thing on this. If I zoom right in on its backside, on its abdomen, and focus, and obviously focus, you can see it's lovely patterning. You can see all the dust that it's got on there. But you can see, first of all, you can see the hairs on its backside. Can you see it's got these lovely straight hairs like that? But if you focus on the little hairs, if I get this just so, And this is at the limit of what this microscope can do. Can you see, sort of here, the hairs are actually branched. They're not just a hair like that, like your hair or my hair. They're more like feathers. It's a straight bit in the middle and then little bits coming off the edges. You have branched hairs. Spiders have all sorts of different types of hairs. They've got sensor hairs, they've got rigid hairs, they've got hairs that they use on their feet specifically for not getting stuck to their own webs. They've got, which are sort of sprung hairs, they've got a kind of a little ratchet mechanism. They've got these branched hairs, and they've just got ordinary straight hairs. So just within something as simple as the common house spider, which is a very much a live house spider, I should point out. And you can see some of the other hairs. These hairs here are for feeling movement when it's sat in its web, and there is pedipalps at the front covered in those hairs as well, so that it can manipulate its prey. So you can begin to see, I hope, why a microscope is such a useful tool for, say, an ecologist or somebody studying whole species like this, because it can give you so much more information and you can begin to discover things about it. And that's really what I wanted to do today. Let me jump out and go to this. Is show you, there it is, in a little petri dish, little glass petri dish. Pop that down there. Another thing that a microscope allows you to do, if you've got a good enough one, is it allows people to identify species, and it allows us to really get to grips with what animals we're looking at. Here are, for example, two little woodlice. Press that button, and I press that button, and I appear in the top there. Right, they don't like the bright lights, so I'll do this quickly. These woodlice are the common, get them in the shot, the common shiny woodlouse, right, which is Aniscus ocellus. I just have to check because I can never remember its name. And the only real way to tell Aniscus ocellus from the common rough woodlouse, whose name I can't remember, is to look at this bit of what is known as its flagellum. The flagellum is the sort of the antenna, and if you zoom in on the antenna, it has, if I get it right, can you see that? It's made up of 3 segments. So there's 1, 2, 3 segments. And the common the common rough woodlouse only has 2 segments at the end of its flagella. And that's pretty much the only way you can tell them apart, is to look at those. They're rather beautiful, I think. And at different times of the year, they look completely different. These, by the way, these white, you can see how it's got these sort of white marks on its back here. This is actually a store of calcium, so that when it goes about its next molt, it has some more calcium waiting to produce the shell, which it needs. It's a divalent ion, and it helps it bind the shell and make the shell rigid. So we can use a microscope, for all sorts of looking at cool creatures like that, interesting stuff like that, and even the simple everyday stuff. Now, during lockdown, normally I would spend a lot of my time kind of going out filming films for the One Show, or zooming all over the country, talking to people and bringing my microscopes to people and letting people actually look down them. Sadly, obviously, that didn't happen. So what I started to do was I started to live stream stuff, and actually you can see the products of all my live streams on my YouTube channel. There it is, Marty Jopson at YouTube. And that's my Twitter address, by the way. I'll show you that later as well. And basically, I just went around the garden pulling out things and looking at things under the microscope and doing the research in advance, because sometimes I had no idea anything. I didn't know anything about woodlice until I started to look at them. So I did a bit of research and came up with stuff. Fortunately, I actually have a pond in my garden. So I was able to look at some pretty cool stuff under my pond. Here's a little bit of pond water that I went out and snagged. I was worried, initially at least, that when I got to this time of year, there would be nothing to see. I could not have been further from the truth. The pond is still teeming with life. Here's some pond water. It's just in a little plastic pot. Duckweed on the surface, pondweed. Each one of this is one plant here. That is one plant. 3 leaves. And then you can just about make out the green roots sort of hanging down. Now, and there are things moving in that water. There are water, there are things alive. In fact, it is absolutely teeming with life. But here's the thing. Different animals, different creatures, different subjects require not just different microscopes, but different types of illumination. Now, a lot of people, when they think about microscopes, and I know it's one of the things they teach you a lot in A-level biology, is to work out magnification ratios and stuff like this. But actually, magnification is not the most important thing for a microscopist. Oftentimes, what's more important than magnification is illumination and how you can illuminate your specimen. Because traditionally, you would look at something under a microscope, and this microscope here is a classic example. And if I show you, if I jump back to this camera here, like this, and I go to that picture. So my camera, I'll turn off all the illumination so you can actually see it. It's so bright it blows out the camera. So there's the microscope. I've got all the lovely lenses and everything, and this button here. Oops, I'm just going to do a thing to stop my camera doing that rather annoying thing there. I'm going to go to that, like that. Boom. There you go. I should have fixed it. Yes, that's fixed it. You've got the body of the microscope here. There's a camera on top here, which is feeding out to the computer, and then I'm streaming it up to you. This is the zoom knob. That allows me to zoom in, and that's the focus. But these two buttons are super important down here. These two buttons control the illumination. And I can have top illumination. That's top illumination. I'll turn it right down so you can sort of see. If I put my hand in there, you can see it's illuminating the top of my hand, but not the bottom of my hand. Turn that off, press the other button, and it illuminates from the bottom up, but not from the top. Now, so that alone is super important. And being able to adjust the brightness is super important. Because when you're looking at some specimens, you want to have light shining through them from the bottom, excuse me, and other specimens, you want it on top. So obviously, when I looked at the spider, I had top illumination. When I look at the head louse, for example, I put bottom illumination on because actually the head louse is pretty much see-through. So I get a better image if I shine the light through it. And sometimes I have a mixture of the two. Now, with this specimen, what you want is neither of those. What you want is illumination coming in at the sides. And it's what's known as dark field illumination. And I can do that by just switching that, turn that off. And that really helps visualize what's inside your specimen. Now, this is a bit of a rubbish little pot. I'm going to move to a different specimen sample chamber in a second, which is made of glass, because this is made of plastic and it's covered in scratches. And what I'm doing now is, rather than illuminating with light that comes up from the bottom like this, shrink straight through the specimen, the light is coming in at an angle like this. So it shines in at a very low, shallow angle. And what that means is, if there is nothing there, if I take this out of the way, you don't see any light. It goes black because I'm viewing it from above here, imagine, and the light comes in at an angle and comes in at an angle and goes out at an angle. So none of the light goes up the tube, so to speak, and makes its way to the camera. So it's black, hence dark field. But if there's anything in the way, that scatters the light, and suddenly all the little things that are in there pop out. So suddenly, can you see all these crazy little critters in here? And if I just zoom in ever so slightly, I don't have to go in very far. I go to sort of there like that. If I just focus up and down, can you see all those little specks of light moving around? Every single one of those, and can you see all those tiny little green specks now? I'm not sure if I have to zoom in a bit more, probably. But all those little green specks, can you see how they're all wriggling around in there? Every single one of those is alive. The little green specks are euglenoids. That one there. That one's a paramecium. Oh, it's just gone through. There's all sorts of other things in there. I can focus up and down. There's some there, I think, are rotifers. I'm not 100% sure. Somewhere in here, there is a mosquito larvae, I noticed. I tell people when I'm doing this show that what I'm going to do is I call it a safari, which is kind of my get out clause, because I haven't got a clue what I'm actually going to show people. Somewhere in here there was a mosquito. There it is. It's kind of gross. There it is. And I have to turn the brightness down on this because otherwise I get too much. There it is. This little mosquito larvae would normally hang. Oh, and there's a bit of duckweed in the way. but it would normally, that's its head there, that's its tail, and it will put that tail. The tail actually has, it's how it breathes. It sticks the tail out of the top of the water and breathes through its tail. It's kind of clever. Let me show you a few things in more detail using dark field, because it allows me to look at these little critters much more easily. Let's have a little look. So what I've got in here is I just separated out, and I'm going to hopefully try and show you one of these. They're called Ostrocons. Here's one. There it is. In fact, there's two there. There's one just zoomed through. This is, come on, an ostracod. It's related to, come on, focus, focus, focus. It's related to, and you can see, these are its feet here. And it's basically, it's related to other crustacea. It's a type of crustaceous which is related to something like the crab or the prawn or the headlouse, the woodlouse rather, that I showed you. And it lives inside two little shells. It's almost like a bivalve. There's another one come through. And this one, I don't know why this is probably one, it's probably dying or something like that, or maybe it's just having a little rest. I don't know. But it lives inside its little shell, and it's got two eyes at the front there. That blob at the back is an egg sac, and you can see its legs, and it sticks its legs out between the shells and wriggles around. Fantastic little creatures, and so much life inside pond water. Okay, that's all good and well. I had in here, I've lost them. There you go. I think that is a nematode of some sort, or possibly an annelid worm. I am no expert on these things, and that's the beauty. I've never seen these before in the sample, and that is the beauty of. And there's a big one there that's sort of staying still. That is the beauty of looking down the microscope at stuff like this. You never quite know what you're going to find. Look at that weird little wriggler. No idea what that is. Could be anything, but it's definitely alive. Look, there goes another one. Zoom! Tiny little worms of some description. You could spend your whole life identifying everything under the microscope, but there's one thing that I always like to show people under the microscope, because it is the freakiest of freaky creatures. It's in here somewhere. Where are you? There you are. It's upside down at the moment. Hopefully it'll right itself. I think what it's done is it's got itself stuck on the meniscus. So let's give it a bit of a poke. Give it a bit of a hassle. This creature here, this is Hydra Viridisima, the green hydra. Now the green, this is related to other medusoids, they're called. They've got these tentacles at one end. It's related to sea anemones and hydrozoids, so jellyfish. Think of the word there. And clearly what it is basically is a little gobbet of jelly, multicellular, and you have here tentacles, and it's flailing the tentacles around trying to find something to eat. The tentacles are covered in what are called nematocysts. Nematocysts are these unbelievably vicious stinging cells, and when something brushes against them, it shoots the sting out. It's like a harpoon, and injects whatever brushes against it with a neurotoxin. One time I was watching one of these little guys, and one of those little ostracods just brushed against it, and must have been injected because it got stuck very momentarily, and then it fell off, and it was dead. And it was just dead within seconds. These things, the toxin is vicious. They're also quite remarkable animals. We think they're immortal. They don't appear to age. We don't know why. Clearly that's of interest. And the other thing about them is, if you think about how, I don't know how much you know about development, but you know that we start as a single cell that divides into two, and we sort of grow and become more and more cells, and eventually you and I are like 30 trillion cells or something like that. But most of the cells in our body, in fact virtually all of the cells in our body, are incapable of producing anything other than what they are now. So a skin cell is always going to be a skin cell. It stays a skin cell, and all it can do is make more skin cells. Whereas some cells, the cells that you've got in embryos, they are what are known as totipotent or pluripotent, which means they are capable of producing more types of cells, stem cells. These guys appear to be made up entirely of pluripotent stem cells, because if you cut one in half, literally, you chop it there, chunk, the bottom half grows a top and the top half grows a bottom. If you cut it into 3 bits, kind of chop, chop, each one of those bits regrows. I mean, obviously it comes to, if you kind of mash it into individual cells, the cells are probably so damaged that they don't regrow. But it's an astonishing, ooh, didn't like that, and it pulled itself back in. It's an astonishing creature. It's about 1 millimeter long, Hydra, and it's green because it has symbiotic chlorella algae inside each of its cells. So photosynthetic algae live inside its cells, about 30 algae inside each cell's microscopic algae that provide it with nutrients. So it's an amazing little bit of a little creature in there, and it's living there in your in ponds in millions. Okay, if it was bigger, it would probably be rather terrifying, given how horribly toxic those neurotoxins are. Right, let's put Mr. Hydra away. It could be Mrs. Hydra. Let's face it, we don't know. Come back to me there. Right. So, okay. That's showing you just a little bit of the sort of microscopy you can do with something like this. As I say, a stereo microscope that's useful for putting big things under, because it's got this big focal distance here, and I can sort of rack it right up here like this. So I can put really big things. I can stick my whole arm underneath there and have a look at things under there. And the great thing about it is I can illuminate things in all sorts of different ways, and that allows me to see things that wouldn't be possibly seen by just regular illumination, sort of straight through or straight above. So low-powered microscopy is just as important as the high-powered stuff, which we're going to move on to now. So this microscope, this is a standard microscope that shoots light up from the bottom. Light comes in at the bottom here, goes through what's known as the condenser here. Let's show you this. I wonder if I can twist this around. I hadn't planned on doing this, so I haven't practiced. Let's see whether I can get that to, there you go. So I've got a standard sort of microscope here. I've got focus, focus, focus. Light source here. There's the condenser there. You can sort of just about see it here. It's sort of a dark black bit here. If I move that out of the way there, ooh, you can't see it. And then on top of that, you've got the lenses here, the objective lenses, and the eyepieces right at the top there, which I peer into. So there's the lenses there, and at the top here, right at the very tippity top, is another one of those cameras that I can see stuff. Now, I'm going to come back down to this like this. So this microscope allows me to do some more interesting things. And one of the specimens I want to show you is, I mean, ironically, we've already seen this specimen. This specimen, we've already seen it in when I showed you the head louse, because that head louse, the reason why the back end of that head louse was sort of dark brown was because it was full of blood, my blood, because that's what it had fed on last, because I combed it out of my hair. I mean, this was some time ago. I should have shown you I'm clean. I want to show you some blood under the microscope. It's dead easy to do. People assume that microscopy, that you have to kind of, it takes weeks and months to prepare microscope images. Yes, sometimes it does. But if you just want a quick and dirty image, you can do it quick and dirty and easily. So let's do that. I've got here a little camera that we're looking at here. I've got some glass slides here. I need 2 for this. And what I'm going to do is I'm going to take a blood sample and put it onto the glass slide, and then we'll have a look at it under the microscope here. And the way you take a blood sample, well, there's all sorts of ways you can do it. The easiest way I find is just to use one of these little jobs. These are these little, it's called AccuCheck Fast Click. You press the button, a needle shoots out, jabs into your skin, and you bleed a drop of blood. It's what folk that have diabetes use when they're going to do one of their tests. I'll use this finger here. So yes, I am going to bleed on your behalf. Here we go. I don't like doing this. If anyone tells you this doesn't hurt, they're slightly lying. There you go. Little drop of blood. Focus on my fingertip. There it is. You actually need a reasonable amount to do this. I mean, somebody's taking a test for their diabetes, they don't need this much. This is way more than I have. You can adjust the settings on this thing. You can change how deep the needle pierces your skin. And I have got it set to the max. Just because I love doing this. No. So you take the glass slide, you put the tiny drop of blood on one end of the glass slide like this. And then you try and stop yourself from bleeding. Put your glasses on. And now what we need to do is we need to smear that blood out over the slide, because as it stands, I have a big pile of blood. The beauty about microscopy as well is you only need tiny amounts of a sample. I don't need a bucket of blood. I need a tiny little drop. So what you do is you take another slide, and this is how they do this when they do a blood sample in a hospital. And this is slightly tricksy. You use a slide and you use capillary action to smear that out across the whole slide like that. And I hope I haven't left that too thick. Ugh. I'm still bleeding, so hold on. Get that. I don't want it, otherwise I'll get blood over everything. So you can see I've smeared the blood out over the slide. I'm just waiting for it to dry ever so slightly, and then I'm going to pop it under the microscope and we'll have a look at what there is to see. Now, this microscope, one of the great things about this microscope is I can put something under the microscope, you can see it, I can see it, we all see it together. Whereas with this microscope, what I have to do is I have to look at it first, and then once I've satisfied myself, which is not dry yet, once I've satisfied myself that I have what I want to see, I then show you. And if I don't, the reason I do it that is because if you don't do that, you tend to make the audience rather seasick with all the sort of zooming about. And what I'm looking for is I'm looking for a patch where I have actually smeared out the blood sufficiently thinly that we can see individual blood cells. And as I said, I was a bit worried that this is a bit thick. Yeah, it is a bit thick. I'll find the best bit I can. Oh, that's not bad there. We could live with this. Actually, here's good. This is good. This is good. This is really thin here. Where's this? It's right over here. This is a good bit. Right. So I'll show you what this looks like under the microscope, just with ordinary illumination. So this is called this type of microscopy is called. Oops, wrong one. This is called Kohler illumination, or technically it's Kohler, because he was German and there's an umlaut on the O. So it's Kohler illumination, Kohler illumination. Basically, all it does is it creates a flat, uniform field of light for to shine through. You can see that those cells, and these are individual blood cells here, they are ever so slightly pink. If you zoom in a bit more, they tend to, and I've gone, and this is a times 10. I'm going to jump up to times 40 now. And you can see they kind of lose their color. And the reason they lose their color is because, oops, focus. We are now looking at them such, individual cells, and there's so little hemoglobin in them, they don't really look like anything. They're just like little kind of round blobs. This is one of the problems, because what we have here are unstained cells. These are just drying out blood cells on a slide. There's no cover slip or anything over them. It's just dry blood cells on a slide. And a lot of the human body and a lot of cells, individual cells, are very difficult to see because there's no contrast. And what you need to do is you need to find a way to create contrast. So see the difference between different parts of the cell, in other words. So at the moment you can see me because my shirt is dark blue and my waistcoat is dark blue and the background is white. So there's lots of contrast that you can see me. These cells, not much contrast. The best way to give cells like this contrast, and the standard way to give cells contrast, is to use a stain. And I don't know if you've used stains yet for your microscopy, but it's a literally centuries old technique for preparing microscope slides. And I'm going to do that. And what we're going to do is I'll switch to this and I'll go to there. so that you can see what I'm doing. So I'm just going to put some gloves on. This stain that I'm going to show you, this is a slightly unpleasant chemicals. It's called a rapid May Grunwald Ginza stain, which is a bit of a mouthful, I'll grant you. And this staining system is designed for use in the field when you're looking for malarial parasites in people. So you take a little blood sample, literally like I just did. You take the blood smear, which is on that slide there, and we're going to dunk it in the 3 chemicals, one after the other, for about the count of 5 each time. And I'm going to go in this way around, because this is the bit that was good for me. So the first chemical that you put it in is this one. This is actually, this is just methanol. 1, 2, 3, 4, 5. Okay. Methanol, and that is a fixative, and that essentially does a couple of jobs. One of the jobs is it glues the cells to the slide. The other job it does is it opens up the cell membrane. It strips away the cell membrane and allows us, the other chemicals, to get inside the cells. So this chemical is called Eosin, and this stains basically the insides of any cell to slightly pink. It basically it's a sort of pretty generic one, and it sticks to cytoplasm. Okay, so anywhere there's a lot of cytoplasm, it will go pink. I've lost count. I'm hoping that's 5 dunks. Again, we drain off. And lastly, into this blue one. Now, this blue one is a little bit like toluidine blue. It's not toluidine blue, and it kind of works a bit like methylene blue as well. There's a fairly common batch of stains, and it's of a sort of a group of stains. And this one specifically targets things like DNA, nucleic acids. So this should light up anywhere where there's a big concentration of DNA. And obviously, that would normally be in the cell nucleus, where the DNA is normally held. Right, where's my water bottle? It's right over here. Let's grab that. Because what I'm going to do now is I'm just going to rinse that off. And it's literally, that's it. Simple as that. 5 seconds in each one, and then wash off the excess that you don't want. Wash, 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 washy, wash, wash. Okay, there it goes. Nice wash. Wash off the back. Made a bit of a mess of this one. This isn't the best stain in the world, because this bit at the top never got stained because I was holding on to it. Put that over there. Tissue. Wipe the back of it. This is where you hope that you're wiping the right side, because it's difficult to know which is which. Wipe back, wipe the front, at least dry that, because ideally what I should do now is allow this to dry off, but hey, I don't have time to do that, so I'm just going to give it a good huff like this, excuse me. Blow as much water off as I can and hope it dries out nice and quickly. Get rid of that, as I say, slightly unpleasant. Actually, the nastiest chemical in those chemicals there is the methanol. Methanol is quite toxic, and that's probably the nastiest chemical. The others are just sort of horribly staining, and they're not very good for you, but if you've got them on your hands, it'll be all right. But it's the methanol you really need to watch out for. Right, I'm going to have another look under here and see how that looks. Good, that looks good. So what I'm looking for now is, yeah, there's a few there. One of the things you learn as a microscopist is your eyes are astonishing. Cameras, rubbish. My eyes can see so much more detail down the microscope when I look down it than your eyes can. And what you see when you see down with a camera down a microscope is you see a relatively small bit of the field of view. I see a massive, great field of view like this. And when I show you the camera image, what you're seeing is this bit in the middle. Okay. So what you can do is I can scan much more area when I'm looking down the microscope. And this is what I'm seeing at low power. If it just autofocuses, I'm seeing that at low power. And you can see immediately that everything's gone dark red, and that's because of that Eosin stain. But I'm picking out in here some little patches of blue over here. And that's what I want to show you. That's what I want to show you up close. So let's go into that. Actually, I wasn't quite centered. Hold on, 2 ticks. Let's get that, at least that in the center. And then I'll try and remember that there's another thing that I want to show you to the left. and then do that. There you go. So we've got our red blood cells now. They're kind of, they're a bit not as thinly spaced here. They're piled up on top of each other. But you can see, there's a couple of things you can see. First of all, they've been stained pink because there's actually a relatively large amount of cytoplasm inside a red blood cell. You can also begin to see the shape of the blood cell. You can see, say, take those blood cells there, that they're actually just donut shaped. The center bit is much thinner than the outsides, and they're flat donuts with a sort of a thin bit in the middle. The reason they're like that is because they have no cell nucleus. The blood cells, they're made in your long bones, in your legs, and just before they're pooped out into your bloodstream, the cell nucleus, because they have one when they're developing, is squished out and comes out. And that, we get rid of that because you need the blood cells to be able to go to the very tips of your fingers and stuff like that. But what I want to show you is that cell there. That cell there, thankfully, it's not a malaria parasite. No, that is a white blood cell. And that type of white blood cell is called a neutrophil. And I know it looks like a sort of a sausage, or a curly whirly, or a what's-it, a blue what's-it. That thing in the middle there is the cell nucleus. And that tells me it's a neutrophil. Just, over this side, there are some other white blood cells. There's one there. Takes a little bit of focusing. There's one just there. And that white blood cell is a lymphocyte. T or B cell, don't know. Probably a B cell, maybe a natural killer cell, don't know. And you can tell the difference because those have their entire cell is taken up with the cell nucleus. Okay, so and if I had time, there's the other. There's the neutrophil. Okay, the neutrophils basically phagocytose. That's the thing. Phagocytosis. They phagocytose mostly bacteria. Actually, they have very little role when it comes to viruses, so they're not really playing much of a role in your immune response to, say, coronavirus. But the T cells and the B cells, that you can see there, that blue one there, okay, they do have a big response in your viral response. So, okay, let's come back to me. I'm running short of time. I want to crack on with this. I'll keep that one just in case anybody wants to have a look at anything else on that. I can go back to that. Right, let's have a think. What are we going to do now? What we're going to do now is I want to show you another technique that people often use in microscopy. And it's a technique called fluorescence microscopy. And to do that, what I'm going to do is, again, quickly prepare a sample. Don't need any gloves for this because I'm using a piece of celery. But I do need glasses on because I'm blind as a bat otherwise. So what I'm going to do is I'm going to have a little look at celery underneath the microscope. It's dead easy to prepare. You cut your celery. I'm just going to cut myself a little chunket of celery. Okay, get rid of that. Don't need that. I've got a little chunket of celery, and I'm just going to slice down here like this and cut a little thin strip of celery. Pop that on my glass slide. I'm just going to check that. Yeah, that looks good. I'm also going to cut a piece of celery vertically down, and I'm cutting through one of the vascular bundles as I do so. That one there, maybe. This is slightly tricky to do. Yeah, that'll do. And then again, I have to cut a very thin slice. You can pop that on my slide like that. Drop of water on that, on each of those. Couple of cover slips. Sometimes it's hard to know when you've got one or two cover slips. Couple of cover slips there, that's one cover slip. Yep, and there's another one. And squash and flatten. Those are quite chunky. And we're going to pop that under here. And I'm going to see if I can see what I wanted to show you. So we'll come back to that and we'll go to that slide there. Because what I wanted to show you is a special technique that uses a different type of light. Normally when we look at something, we're looking at it with ordinary white light. This is good. There, that's fun. But what this microscope can do is get the settings right. Yep. And I'll go to that one, and I'll go to that one, and I'll go to that one. There. Right, so this microscope allows me to do more than just look at light shining up through it. What I'm going to do now is I'm going to use what's known as fluorescence microscopy. Now, fluorescence microscopy is actually the sort of techniques that people are using in laboratories right now out there doing science. And it's a very powerful technique because it allows us to illuminate things and to show up individual molecules and individual types of or structure within cells and within side structures like this. So what we have here, this is the vascular bundle in this bit of celery. So if I break this bit of celery here like this, and then pull back like that, you get the bits of string from the celery. Here's a celery string, okay? That thing there, okay? This is one of the vascular bundles, okay? The vascular bundle contains the xylem and the phloem inside it, and they're the sort of the stringy bits in celery. And what I've got here is I've cut through the vascular bundle, and you can just about make out individual cells. So this here is what's known as parenchyma. This is just the body of the cell. This here is the phloem, and this bit here is the xylem. And it's quite difficult to see what's going on. If I turn on my fluorescence microscopy, we can see what's known as autofluorescence. So what I'm going to do is, rather than illuminate it with light from below, I'm going to shine a light on, and I'm going to go to my desk camera so that you can see. I'm going to explain on the microscope. So normally the light comes up from the bottom through the specimen, and we see, well, actually you can see the image there in the top right hand corner. What I'm going to do now is I'm going to illuminate with some LEDs, some ultraviolet LEDs that are at the back here. The light shines all the way along through here, hits a mirror just in here, and then bounces down to the specimen, right? At which point, some of the specimen will absorb that ultraviolet light. It will then fluoresce. It will emit that ultraviolet light again, but at a different wavelength. And this time it'll emit it it'll shift the wavelength from ultraviolet into the visible spectrum, so we can see it. Some of that light comes back up and goes into the eyepieces. It's all rather complicated, but the end result is very pretty. Let me show you what you can see generally. So I'm going to turn on the ultraviolet light. All I have to do is that. And there it is. So what you've got there is suddenly it goes black because we've turned off the ordinary lights. We've only got ultraviolet light. And you've got this sort of weird greenish glow coming from, this was the xylem. But I want you to look at all around the phloem. The phloem was this bit here. But around the phloem, hopefully you can see that, it's sort of dark on dark. But around the phloem, there's this sort of faint halo of little red dots. Those little red dots are chloroplasts, because when you illuminate with this particular wavelength of light, it makes the chlorophyll glow red, which is a bit counterintuitive, but that's what it does. Not green, but red. So every single one of those tiny little dots here is a chloroplast. And you'll see there are no chloroplasts inside the phloem, but there is a bunch of them just outside the phloem. If I change the wavelength, I illuminate a different type of molecule. Specifically, I'm now illuminating lignin, and anything that's made of lignin is glowing blue. And the vessels in xylem are reinforced with lignin, and that's now glowing up blue. But what I really want to show you is the cross section. So I'm just going to go back to the desk to this picture whilst I have a look at the cross section, because hopefully I've managed to section through. Yes, I have. Good. Some of the Xylem vessels. So I sectioned down through the stringy bit, through the vascular bundle, and what you can see there are the individual Xylem vessels, but now you're seeing them in sort of longitudinal section, right? So it's a bit out of focus, but can you see there's one there that runs all the way down there? So there's the top of it, and there's the bottom of it, or the other way around. But what I want you to see is the fact that these Xylem vessels are reinforced with great long spirals of lignin. Can you see here? It's like an enormous, it's quite good just there. It's like a great big spring coiling up through the tissue of the plant. And the only reason you can see that, look, there's that's got, oh, look, oh, that's a really good one. That's a really good one there. Look at that. Oh, that's brilliant. Yeah, look at this bit here, all the way down there. One Xylem tracheid vessel with a spiral of lignin running all the way up it, illuminated using what's known as autofluorescence. And that is one of the beauties of microscopy. You can use it to show fantastic things like this. Right, I've talked for 5 minutes more than I was supposed to talk, which means we're going into Q&A time. So let's come back to this picture. Let's go to this picture. It's a little bit more close up, a little bit more intimate. I'm up here for you. So we ended on a bit of celery and some autofluorescent microscopy. We started by just looking at spiders and stuff like that. I hope that's sort of given you a bit of a view of different types of microscopy, and I've tried to put in as much science as I can so that you guys get a chance to actually see some stuff that you wouldn't normally with these fantastic microscopes. So, okay. If you've got any questions, please post them in the chat below. I will attempt to answer those questions. Let's have a little look. We've got questions here coming up. The first question is, what inspired me to become a scientist? When I was a kid of about 8, I think, my granddad was a member of something. It was this thing called the Reader's Digest. I don't know if it still exists. Basically, what they do is they send out a little magazine, yay big, once a month. And that magazine contained inside it short stories and stuff like that. Hold on, I've just got to do something here. Is that? Yes, that's stopped. Good. Contained inside it short stories and bits of interesting information. But they also used to sell these books of science. They'd be kind of like, kind of this really big hardback books of kind of birds of Britain or animals or inventions. And they were what really got me into science. And I used to go to the Science Museum in London a lot with my dad, because that's where I was brought up. And that's really what got me into science. So it's kind of those two folk really did that for me. Why doesn't the blood look red under the microscope? Okay, imagine that each blood cell is 8 micrometers across, 8 microns, 8/1000ths of a millimeter across. Clearly, its volume is tiny. The amount of hemoglobin inside the blood cell is therefore equally very, very small. It is, oh, something has quit. I've just had a few, are you still seeing me and hearing me? I hope so, because I just had an error message come up. Maddie, just everyone can still see me. Good. So yeah, each one of those red blood cells is teeny tiny. So there's very little hemoglobin in there. So when you look at one red blood cell, it just doesn't look very red. Equally, with the amount of light that I'm blasting through those red blood cells, it kind of washes out any of the color. So you only really notice that you have a whole load of, that you have red blood when you pile them up in millions, or I don't know how many blood cells are in a single drop of blood, billions probably, and then it appears red. It's only when you have all of those tiny little drops of blood piled up that you see the redness. Does it matter if each chemical is mixed or cross-contaminated? I assume we're talking about my my stain system. So the stain this lot here, the 3 chemicals? No, not really. The whole point of this stain system is it's designed to be super duper easy to use anywhere, literally in the field. So the idea is if you're testing for malarial parasites, you get a set of this stuff. It doesn't need to be kept in the fridge. It's room temperature stable. You take a drop of blood off somebody with a little lancet like this thing here, and these cost pennies. You put the drop of blood, smear it out just like I did, dunk, 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 washy, wash, wash, stick it under a microscope, boom, you can see, and it doesn't have to be very high powered, it doesn't have to be a super duper clever microscope, and you'll be able to see immediately if you have malarial parasites in the blood. So it's a super easy way of detecting blood parasites that can be used pretty much anywhere, because it can be used you just want somewhere slightly clean with some clean water. And that might be hard to come by. So it doesn't really matter if I kind of dunk from one to the next, to the next, to the next, and some of the chemical gets across, because in the end, it doesn't matter. I mean, one of the key things about this technique that is a problem for microscopists, and this is what we've moved on, we don't really we don't use staining much these days in modern microscopy for sort of research purposes, because staining, you have to kill the cells. You have to break open the cells so that the stain chemicals can get into the cells. And you normally do that by ripping apart the cell membrane, and obviously that kills the cell. And the methanol then cross-links all of those cells, the proteins within themselves on the surface, and stops them getting washed off. It also glues them to the cell, the surface of the slide, so they're actually literally stuck down, which is why you don't need a cover slip. You just dunk, 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 and it's fine. What inspired me to be a scientist? I've answered that one. How did you keep the spider still? Spiders are pretty well behaved, actually. They get a bit upset if it gets quite warm under the lights. So they do tend to get a bit fritzy after a while, and they start running around. Tenegaria Domestica, common house spider, is pretty good. As long as you keep it in the dark, pull it out, put it under the microscope, look at it, put it away again. And I always use glass. So the spider here is in a glass dish, because the glass is just, you can use plastic there, that you can see it. I don't want to tip it over or shake it. There he is. Or she, I should say, she. Squishy the spider. I use glass just because you can see through better. Put that down gently. Don't want to scare it. Some spiders are much more jittery. I like to show people, there's a lovely spider that you can find quite often in the summer called the zebra jumping spider, which is beautiful. It's much smaller. It's only a couple of millimeters long, and it's a jumping spider, so it literally jumps like this, and it kind of skates around. It has 2 huge forward-facing eyes, and then 6 more smaller eyes. Like literally, it's kind of dinner plate eyes at the front like this, so it looks quite horrific when you look at it down the microscope. It's got these enormous chelaceae, the fangs at the front are kind of huge things sticking out. But it's a sweet little spider, and it's also zebra striped, black and white striped. It doesn't have forked hairs on its bum though, I noticed. What types of microscope? Tell me about them. They want to know more about the microscope, the types. Okay, so first of all, as I say, Zeiss makes these microscopes. So these are professional level microscopes. They're sort of research level microscopes. This one is a transmitting light microscope, but it's also got all sorts of other capabilities. It's got the fluorescence on it, so I can do fluorescence microscopy, or technically epifluorescence. I've got cross-polar microscopy I can do on this, so I can do, which is less to do with biology and more to do with material science. I can do phase contrast, I can do dark field on this, I can do all sorts of clever things with this microscope. And as I said earlier on, oftentimes it's not the magnification that you're wanting that's the clever thing, it's how you can illuminate your specimen. And with this microscope, I've got so many different options, it enables me to see really small things in a way that you couldn't otherwise, because as I said as well, a lot of very small things, there's no contrast, and it's very difficult to see them. Phase contrast is a clever way of doing that. I didn't have time to show you phase contrast. But there's that. That's that microscope. This is a stereo microscope, as I said. The reason it's, and what actually, rather than having one light path, it's got two light paths that go all the way down to the lenses. It's got two sets of lenses that then sort of diverge like that, so that when you look down, it's like you're looking at your specimen from two different angles, with one from each eye. So you actually get kind of like a 3D effect as you scroll up and down through it. It's very clever, and gives fantastic imagery. You don't get to see that with the camera on top, though. So this is what they used to call a dissecting microscope, because you could dissect things underneath it, because you could put a whole thing underneath it. Right, we are just about at the end. If there's any more questions, you can send me them, and if you want to see more microscopy, go here and look at my Micro Minutes series, and or follow me on Twitter and send me questions on Twitter. If there's anything you want. With that, it's just gone 7, so I'm going to draw a veil over this wonderful microscopy. I hope you enjoyed that. I hope that was vaguely illuminating. Thank you very much for watching. My name is Marty Jopson, and I'll see you another time, hopefully. Bye-bye.